Assalamualaikum warahmatullahi wabarakatuh everyone, and welcome to Science Story. My name is Nurashida Miti Abdul Rauf and I'm gonna be your storyteller for today. So today, we're going to talk about DNA recombinant technology of silkworm silk and spider silk as potential bioactive wound dressings and skin grafts. Okay, so first, we're going to talk about silkworms. Silkworms are the offspring of moths. They spew out thread from the tiny holes in their jaw, which they use to spin into their egg-bearing cocoons. This entire production takes about a mere 72 hours for it to form, during which time they produce about 500 to 1,200 silken threads. There is silk fibrin that gonna be retrieved in sufficient quantities from the cocoons. However, the silk fibrin that we retrieve from the silkworm cocoons do not have bioactive functions. Meanwhile, on the other hand, spider silk proteins can be produced in fusion with bioactive protein domains, but the recombinant procedures are expensive and large-scale production is challenging. So, the best win-win solution is definitely by combining these two types of silk to fabricate affordable and functional tissue engineered constructs for wound healing applications. Okay, so first, what kind of wounds or what categories of wounds that is suitable for this kind of bioengineered applications? Skin regeneration is an important challenge due to complexity of different kind of wounds as well as costly treatment. Wounds like diabetic foot ulcers, venous ulcers or pressure sores are chronic wounds where the healing process stalls due to the pathophysiological conditions. Around 6.5 million people are suffering from chronic wounds with an estimated cost of 25 billion USD. Increasing numbers of such cases impose a great demand of effective solutions for healing with low cost. Other traumatic wounds like full thickness burn wounds and lacerations also impose a great threat due to the significant skin loss and damaged tissue integrity. Use of autografts, artificial grafts, polymerase dressings, tissue engineered cellular grafts and other therapeutic approach have resulted in improved wound healing outcomes. However, the limitations of available grafts and wound dressings necessitate advancements in the current technology. Another challenge in therapeutic approach in treatment of chronic wounds is to combat cellular senescence, bacterial bioburden, and prolonged inflammation. Therefore, there is a great demand of bioactive wound dressings or grafts which not only stimulate cells for proliferation but also provide antibacterial action and therefore improve the wound healing process. The strategy of this DNA recombinant is to functionalize silk fibrin bulk material using spider silk assembly without any chemical reactions to avoid any toxicity associated from the reagents and byproducts. Two types of silkworm silk fibrin is used in order to look further into the development of stable functional grafts. Bombix mori, the mulberry silk variety, is available worldwide and has been extensively explored as a biomaterial for the tissue engineered application. And the other one is Antheria astama, a non-mulberry silk variety, also possess excellent biocompatibility and has great potential in tissue engineering, although less explored. Silk fibrin from both silkworms hold same properties like biocompatibility, low immunogenicity, biodegradability, and tunable mechanical strength, which is essential for developing 3D engineered tissue products. The ease of processability and flexibility also allows fabrication of various formats like films, sponge, nanofibrous mats, hydrogels, microparticles, or nanoparticles. However, for the DNA recombinant being discussed here, the format used are nanofibrous mats and 3D porous scaffolds in order to develop bioactive foam dressings and cellular dermal epidermal skin grafts respectively. Basically, side-fibrin-based substrates are used in bulk material, which are top-coated with 4 wrapped CT proteins fused to a cell-binding motif from fibronectin containing the RGD sequence, a growth factor which is a fibrillase growth factor FGF2, or cationic peptides with antimicrobial properties AMP. RGD in blue color and FGF in brown color enhance cell binding activity and cellular growth. Meanwhile, the AMP in grey color prevents bacterial growth and biofilm formation. The cell assembly between the spider cell and the subfibrin substrates thus lead to the fabrications of bioactive materials with potential as foam dressings and bilayer skin substitutes. Cellular behavior on the coated mats was examined through antibacterial and cell proliferation assays and morphological observations. Further, 3 types of skin cells were cultured on the scaffolds to develop a mature skin substitute in vitro. The cellular process was observed by examining the proliferation rate and specific cellular stainings. The results reveal an easy, efficient, and affordable way to develop functionalized constructs for tissue engineering applications. conclusion, multifunctional mates with antibacterial and cell recruitment properties may, in short, make them a possible candidate for the treatment of chronic wounds. Due to favorable physicochemical and biological properties and its compatibility, silk fibrin serves as an acceptable bulk material for tissue engineering scaffolds to enable recombinant spider silk to self-assemble on it. Using co-culture of the skin cells in the spider silk-coated silk fibrin scaffold, dermoepidermal grafts were easily grown. The present study reveals a simplistic approach to the manufacture of affordable tissue engineered products given the high availability and flexibility of silk fibrin bulk material and the different bioactivities that can be generated by coating with spider silk fusion proteins. Okay, so guys, that's all for today's science story. How do you all enjoy it? Yeah, thank you. Till we meet again. Bye.